many of you may not know that actually congenital heart defects
Viele wissen vielleicht nicht, dass angeborene Herzfehler
are by far the most common type of birth defect in babies.
die häufigste Geburtsfehler bei Babys sind. Weitaus die häufigste.
So almost 1% of all babies born have some kind of a heart defect.
Fast 1% von allen Babys, die geboren werden, haben irgendeinen Herzfehler.
So this is a picture of one of the most severe heart defects that I study.
Das ist ein Bild von einem der schlimmsten Herzfehler, die ich studiere.
It's called hypoplastic left heart syndrome, or HLHS.
Das heißt hypoplastisch Linksherzsyndrom oder HLHS.
And it occurs in about 3% of all babies that are born with some kind of a heart defect.
Und bei 3% von allen Kindern, die einen Herzfehler haben, sieht man diesen Fehler.
And remarkably, in Jacobsen syndrome, about 5 to 10% of all infants are born with this heart defect.
Und bei Jacobsen ist es 5 bis 10% von allen Kindern, die mit diesem Defekt, diesem Herzfehler geboren werden.
So on the left is just a cartoon. I don't know if my pointer is showing. This is just a cartoon on the left of a normal heart.
Links seht ihr eine Abbildung von einem normalen Herz.
And I think you can appreciate on the right
Ich glaube,
the
dass
difference.
dass ihr den Unterschied sehen könnt, mit was man zur rechten Seite sieht. Der größte Unterschied ist, dass diese Kammer zu klein ist.
In addition to that, the other structures on that side of the heart are small and affected as well, usually.
Dabei sind normalerweise die andere Sachen an dieser Seite des Herzens auch zu klein. Da sind die zwei Klappen,
Those are usually abnormal as well.
die Mittelklappe und Aortalklappe, die sind normalerweise auch
And you can
abnormal.
also see that the main blood vessel that gets blood out to the body, called the aorta, is usually very small as well.
Und die Aorta, das ist die Hauptader, wo das Blut Richtung Körper geht, die ist normalerweise auch zu klein.
And the other thing I think you can appreciate is it almost looks like this wall has thickened and grown inward
Und
to make that chamber small.
es sieht eigentlich so aus, als ob diese Wand zu dick geworden ist und nach innen gewachsen ist und dass dadurch den Herzkammer zu klein ist.
So that might suggest that there's actually too much growth of that heart tissue in this disease.
Also das könnte darauf hinweisen, dass es eigentlich zu viel Wachstum gibt von diesen.
Does that make sense to everybody?
Dass dieses
So
Herzgewebe
too much growth
eigentlich zu
inward
viel
maybe causes
nach
that very
innen
small
wächst
chamber.
Und dass darum das Herzkammer zu klein ist.
Like I show here with the arrows.
Das zeige ich hier mit dem Pfeil.
But like most things in medicine, it's not that simple.
Aber so einfach ist es natürlich nicht wie immer in Medizin.
And I hope you don't have a sensitive stomach, but all of these are different versions of hypoplastic left heart syndrome.
Hier sieht man verschiedene Versionen von hypoplastischen Linksherzsyndrom.
So if you look, for example, at the upper left heart,
Links oben zum Beispiel,
you can see that that wall is massively thickened.
da sieht man, dass diese Wand viel zu dick ist.
And then you can see that little white circle of what's like scar tissue.
Und da dieser kleine weiße Kreis, das ist
And then inside of that is the very small cavity or chamber volume itself. And then on the left bottom is another version where you see something very different, where the ventricle is just very small all the way around. And you don't see that scar tissue like you do in the upper left heart. So sometimes we call this version on the lower left a slit-like left ventricle. And in Jacobsen syndrome, most commonly we see the slit-like left ventricle version. And in that version, the valves are actually completely blocked off. They do not open at all. So you could think that in a way that may be the most severe form of all these. So we're trying to understand what is actually the mechanism that leads to such an abnormal heart like this one here. So when we take a little sample of this heart and look under the microscope, this is what we see on the right. And those are just, like I alluded to before, heart cells called cardiac myocytes. And those are the working cells, like muscle cells, that actually make the heart contract. Question? Oh, sorry. So hypoplastic left heart syndrome is the most common cause of death in all infants born with congenital heart disease. And as I mentioned yesterday, it now looks like it's probably the most common cause of death in people with Jacobsen syndrome. In the United States, there are about 1,000 or 2,000 babies born every year. And I estimate that it's probably costing directly over $1 billion a year in the United States to take care of these babies. There is no cure for this disease. But what we do is, over a period of a few years, with 3 open heart surgeries, starting from right after birth, we basically convert the heart to be what we call just a 2-chamber heart. And as a result, the right ventricle, whose job is normally only to have to pump blood to the lungs, now is used to pump blood out to the body. And while this has been a great step forward to allow these babies to live, this first approach was done about almost 50 years ago now. But we now know that the right ventricle will likely fail over time. And so many of these people will eventually need a heart transplant. So one of the areas of controversy in this field has been trying to understand what actually causes this defect or collection of defects. So one theory is that if there's something that blocks blood coming into the developing heart, that will prevent the heart or the ventricle from developing normally. And some people will refer to that as the no flow, no grow theory. And there's actually now some nice evidence in a mouse model to show that if you actually block flow into the developing heart, relatively late in heart development, you can get one of those versions that I showed you of hypoplastic left heart syndrome. The other theory, which I think specifically applies to Jacobsen syndrome, is that there's a genetic change that affects heart development very early on. And if we ever want to have a way to ideally prevent this heart defect, it would be very important to know when these defects, when these problems are occurring during heart development. So until very recently, one of the problems has been there have not been good animal models to replicate this disease. And when people have tried to study heart tissue from a hypoplastic left heart, it's very difficult to know how to interpret those findings because you're looking at something that happened where the actual disease process occurred much earlier. So this is the little boy that I referred to yesterday that I first saw in July of 1995. So he has Jacobsen syndrome. And this was in his chart when I was taking care of him, showing the obvious problem with his chromosomes. And so back then, even though I had no idea how, I figured there must be a gene in that region that is important for heart development. So as I mentioned yesterday, through the conferences like this and talking to so many of you, we learned a lot about Jacobsen syndrome. And that led to this paper that we published a long time ago now. So we learned some really interesting things about heart disease and Jacobsen syndrome. One thing we learned is that about only half of children with Jacobsen syndrome have any kind of heart defect. So even if you have two people with identical deletion size, one could have a completely normal heart and the other could have hypoplastic left heart syndrome. And we call that incomplete penetrance. Meaning there must be other factors that can affect whether or not you actually get a heart defect. And as I mentioned yesterday, there's a very high frequency of this in Jacobsen syndrome. And out of those 1,000 to 2,000 babies a year in the United States, maybe about 1 to 2% of them have Jacobsen syndrome. And the other thing that many of you will appreciate is that many of the other common heart defects that occur in the general population also occur in Jacobsen syndrome. like a VSD, for example. So how many of you have a child that's had some kind of a heart defect? Yeah, it's more than half. Yeah, so born out. Okay, so we've talked about this concept of mapping the deletion called the microarray. This is kind of an old version of what a microarray is, but it's an incredibly powerful tool to define exactly where the breakpoint is. And once you know the breakpoint, you can know exactly which genes are affected. And with that information, we were able to narrow down a very small region So the blue shows the smallest terminal deletion of that little girl yesterday. The one that died after her heart transplant, sitting next to the bunny rabbit yesterday in that picture. And rarely we have come across other patients with heart defects as well, shown in the red, that have what we call interstitial deletions. And so if you look at like this patient with that patient, hypoplastic left heart, there's just a small region of overlap. And that small region of overlap only has these genes, 6 genes. And already we had a pretty good clue that one of the genes, the one on top, plays a major role in heart development. All right, so I've just summarized now about the first 10 years of my work in about 10 minutes. $1 million like that. Does that make sense to everybody? So that's how we got to this point where we think we found the heart gene out of that big chunk that's gone missing. Any questions? Go ahead. I don't know, I kind of like running back and forth. Is there a laser? What's that? Is there a laser? All right, it won't get recorded. Okay. That might be a good thing. All right, I'll try to point. That's okay. I can use the pointer here, I think. Okay, all right. Moving on. So we study heart development in the mouse because it turns out the mouse heart is very, very similar to a human heart. Got one? All right, on cue, perfect. All right. So the first thing we wanted to do is to find out where this gene is working as the heart is developing in the mouse. And it turns out that the heart actually has a lot of different types of cells that are required to make up all the different structures. So for example, the cells that make up the valves are different from the actual muscle cells that are the pumping. So all this shows you here is an experiment where the blue is showing where the ETS1 gene is expressed in a developing mouse heart. And this blue coincides with two different types of heart cells called the neural crest. And that's the blue down here. This is neural crest, so you can see how that's the same as that. And the other population of cells is the inner lining of the heart chamber called the endocardium. And that's what's shown here in brown. So it turns out the endocardium, these cells, have a very critical role in regulating how the ventricles actually develop. And I'll come back to that point in a few minutes. The other population of cells, the neural crest, I'm going to tell you about now, but keep one thing in mind. Interestingly, even though I just showed you that in at least one form of hypoplastic left heart syndrome, the heart muscle cells are probably abnormal because there are too many of them. But clearly, we've shown that ETS1 is not expressed in those heart cells. So it suggests that it's the loss of ETS1 in one or both of these populations of cells that is what causes all these different heart defects that many of your children have. Does that make sense? Any questions? All right, so moving along, most of the rest of what I'm going to talk to you about is our studies on what ETS1 is doing in these two different cell populations, okay? So this is a cartoon of cardiac neural crest cells. And cardiac neural crest cells are really an amazingly complicated and beautiful population of cells. And as the embryo is developing, these cells actually start out not in the heart, but actually in the neural tube, which is the future spinal cord. So within neural crest cells, there are subpopulations of neural crest cells. I'm going to tell you about the subpopulation called the cardiac neural crest cells. And as you can see here, these cells have to migrate from the neural tube in a very precise manner to form specific structures of the heart, including what I'm going to show you in a few minutes is the so-called septum that separates the blood that goes to the lungs versus the blood that goes out to the body. Now, the other day, we were talking about why so many children with Jacobsen syndrome have red hair. It turns out there's another subpopulation of neural crest cells that affect skin color and hair color. So when we get rid of ETS1 in neural crest cells, which I'll show you in a moment, the mice that normally have gray fur color have white fur color. So that's why I think it's probably this gene that's responsible for such a high frequency of red hair. So again, just to summarize, this is an incredibly beautiful and precise process that allows these cells somehow to know where to go to do what they need to do to form these structures of the heart, okay? And there are a lot of other groups around the world that have been studying neurocrest biology as well. So as I mentioned before, we hypothesized that it's the loss of ETS1 in one or both of these populations of cells that will cause the heart defects in your children. And I must say, unlike any other talk I give, it's such a personal experience to connect with you, to say it's your children that we're talking about here. So what I'm first going to show you is what happens when we use some genetic tricks to actually specifically eliminate the ETS1 gene in neural crest cells. So I hope you can see this okay. On the left is a mouse heart that's fully developed. And this is actually a cardiac MRI of a mouse heart. So this is the normal left ventricle. That's the normal right ventricle. And in the red is the aorta, which you can see here is normally connected to the left ventricle. And it's a little bit hard to see, but the pulmonary artery is connected to the right ventricle. And you can also see here, this black is the wall that separates the left from the right ventricle. That's called the ventricular septum. So if you have a hole in that septum, that's called a ventricular septal defect, or interventricular septal communication, IVC sometimes. Now in the normal heart, the aortic valve, shown here, has three parts, or what we call three leaflets. And normally in the mouse, those neural crest cells help to form these valve leaflets. And the septum, as you can see down here a little lower. So the blue in this slide is showing you neural crest cells in a normally developing mouse heart. And this is just another way to show the same thing, except now the neural crest cells are red. So now what happens when we eliminate ETS1 in the neural crest cells? Now we see something very abnormal. Compared to this, we see a hole or a communication between the two ventricles. So that's what we call the VSD or IVC. And if you look carefully, you see how the aorta is aligned with the left ventricle there? Here it's aligned with the right ventricle. So we call that double outlet right ventricle. We also found commonly a problem with the aortic valve. Notice how there's almost no blue. And instead of the normal 3 leaflets, we just see 2 leaflets. And this is showing the same thing. Very few red dots compared to that. And again, the very abnormal valve leaflets. So what we wanted to do is try to figure out, okay, now we've shown it's the neurocrest that can cause this type of heart defect. And the question is, how does that happen? So the top here is a cross-section through a normal mouse embryo as the heart is developing. And a close-up within this box shows these cells, which are the neurocrest cells, are normally moving in a very specific way to the heart down there. And what we see in the mutant mouse that does not have the ETS1 gene is a disruption of that normal process of what we call cell migration. So in the future, we're hoping to understand exactly what the genes are that ETS1 is regulating to cause this cell migration defect. Does that make sense to everybody? So it's affecting how these cells normally move to that part of the heart. And just like in human patients, in some genetic strains of mice, we see a heart defect, but not in others. So in one strain in particular, that's when we almost always see this heart defect, okay? But that doesn't answer the question about hypoplastic left heart syndrome, right? Just to review what I said before, we think in some cases it may be due to this actual overgrowth of heart cells. So that's what I'm going to spend most of the rest of my time talking to you about. All right, so it turns out, amazingly, before I even really got very far along with this, there was a paper published in 2003 describing what the ETS1 gene does in fruit fly heart development. And it may seem incredible to think that we have something in common with that fruit fly that you kill every time you see one. But in fact, what many people have appreciated now is even though physically a fruit fly is very different from us, they share many of the same critical genes required for heart development. So on the left is the heart of a developing fruit fly embryo. And what you see here hopefully is green cells and yellow cells. And even in the fruit fly heart, there are two different cell types that make up the functioning heart. So this is basically just a tube with one cell layer around it. And what this group showed in 2003 is that when you have a mutation in ETS1 called pointed, you get, compared to that, a very abnormal looking heart. Everybody buy that? And remarkably, what you really see here is an excess of heart cells. And when I first saw that, I thought, wow, that's incredible, because that is similar to maybe what we're seeing in some of these human hearts. And much more recently, we could do in one of my collaborators' lab an experiment where we just eliminate ETS1 in the cells that give rise to those heart cells. So not the whole animal, but just specifically in those heart progenitor cells. And even with that kind of a specific knockdown, we see something, again, very similar. Namely, an excess of these different types of heart cells. So basically, what I've shown you, in a way, is hypoplastic left heart syndrome in a fruit fly, we think. Don't worry about this busy slide. All I want to point out to you is that even in the fruit fly, it is a very complex series of steps involving many genes for that even very simple heart to develop normally. And it turns out, if you look at these three genes, this gene is called Tin Man. This is ETS1, our gene. And this is another gene called Notch. And what I didn't tell you before is that in human hypoplastic left heart syndrome, there are only maybe 5 or 6 genes that are known to cause hypoplastic left heart syndrome. And Tin Man, ETS1, and Notch, amazingly, are 3 of those genes. So to me, this is a hugely important observation, because it shows how these genes are extremely important and highly conserved. And it means that we should continue to study these genes in fruit flies, because as you can imagine, there are great advantages in studying a fruit fly even compared to a mouse, let alone humans. And one of the things that's apparent on this slide is that all 3 of these genes seem to be regulating another gene called even skip. Now, we know that even skip is a gene that's critical for, in general, embryonic development. But up until now, no one really knows what its role is in heart development. But what we could determine, thanks to our collaborators from the Mayo Clinic in Minnesota, is that they have identified at least 1 patient with hypoplastic left heart syndrome who was found to have a mutation in this gene. So we are actively studying what this gene is doing in normal heart development. But I think this is a very exciting example of how our research has led to discovering a new gene. In a patient that does not have Jacobsen syndrome. So again, we are studying now what even skip is doing in normal heart development. All right, so I want to show you some other more recent data on what the ETS1 gene has been shown already to be doing in heart development. So don't worry about this slide. It's very busy, but I'll walk you through it. In this case, this is a picture of the heart of a frog. This is a normal frog ventricle. That's all you need to know for this. And in this case, my collaborator from the California Institute of Technology, or Caltech, showed that if you eliminate ETS1 in what we call the cardiac mesoderm, which is the population of cells that give rise to heart cells, look what you get here. Compared to the normal ventricle, you see a smaller chamber and an excess of heart cells. Everybody see that? And so again, this same theme keeps coming back about extra heart cells. And taking it one step further, not only do we get extra heart cells, but remember I was telling you about the endocardium, that population of cells that lines the heart? This is now a picture showing, stained in green, endocardial cells in the normal frog heart. And now in this heart, in which ETS1 has been eliminated, there's almost a complete loss of those endocardial cells. And with what appears to be, again, increased cardiac myocytes. So what it appears is that ETS1 is regulating two different populations of heart cells, so that when you lose ETS1, you lose one population of heart cells and you gain more of the other population of heart cells. And since we think this is due to having too many heart cells, we actually looked at some genes that we know are involved in heart cell growth or proliferation. And all you have to see is that compared to a control in blue, when we knock out ETS1 in that cardiac mesoderm and get all those extra heart cells, a few of these genes that we know are involved in heart cell growth and proliferation are massively now overexpressed. So this fits our model that in some cases it's too much heart cells causing that heart defect. Can we maybe turn down the lights again one more time? Because this is a really cool slide that I want to show you. So this is an experiment done by someone named Shui Ni, who worked at Caltech initially. So the red is just a stain for normal heart cells. So again, that's the normal frog ventricle. And here it is stained in red, like a section through it. And when you knock down the ETS1 gene in the cardiac mesoderm, you get that abnormal ventricle. And here, compared to the control, it just looks like a small ball of heart cells, right? And we think it's because we eliminated the function of ETS1 in that population of cells called the cardiac mesoderm. So Shui did a really cool experiment to prove our hypothesis. And what she did is she took a heart that was meant to be this, meaning a mutated heart, and she grafted, so she just basically attached to this heart normal cardiac mesoderm tissue. And for whatever reason, whatever is contained in that cardiac mesoderm tissue, it was able to prevent that heart defect. And I'm sorry I don't have a picture to show you that in this rescued heart, we now have gained back those endocardial cells that were lost in this heart. So together, all this suggests that it's the endocardium that has a critical role in regulating the ventricle. I think I'm going to skip this slide because I've basically just said everything. All right. So we wanted to take that one step further. And if our hypothesis is right about the role of the endocardium, then there was one more genetic experiment that we could do to prove that. So this is a normal mouse embryo about halfway through gestation. And at this point, this is the developing left ventricle, and this is the developing right ventricle. And if you come a little more forward towards you from the screen, this is the right ventricle and basically like the artery coming out. So using some genetic trickery, we could actually kill off the endocardial cells before they can even begin to function in heart development. And when we do that, we see an extremely abnormal heart. Compare that to that. This is the same scale, by the way. And these embryos died like a day later. All right, so what we think is all this evidence points to a critical role for the endocardium as one mechanism that can cause hypoplastic left heart syndrome. And so Lu Wang in my lab is studying all of this. And a more recent experiment he's done is to begin to look at what the other genes are that ETS1 is regulating. And not surprisingly, we find a lot of genes, including these 12 genes or so here, that ETS1 is directly regulating. And if you look at what the functions are of those genes, it makes complete sense. And very interestingly, it's already been shown for some of these genes, like this one, that a mutation in this gene can cause a different type of heart defect that can also happen in Jacobsen syndrome. And you have a question? She needs a microphone probably, right? Or if you want to say it, I can repeat it. Yeah, so that is, the endothelium is the group of cells that includes the endocardium and also cells that make up blood vessels. I didn't want to discuss it, but there's also a major effect on the development of the coronary arteries, and so that fits very nicely with this. Does that answer the question okay? All right. All right, so just to summarize, we know that ETS1 is expressed in the endocardium / vascular endothelium and neural crest, but not in actually those working heart muscle cells. And that in early heart development, ETS1 looks like it regulates cell fate determination. Specifically affecting this balance between endocardial cells and cardiac myocytes. And those last studies that I showed you suggest that it may be possible for early intervention to rescue that very abnormal hypoplastic left heart ventricle by being able to restore or preserve the endocardial function. Another set of experiments that we're getting ready to do is to eliminate the endocardium at later stages of heart development. Because one possible explanation for those different types of hypoplastic left heart syndrome may be exactly at what time in heart development the endocardial function is impaired. And those experiments in my lab are underway right now. And finally, knowing what the mechanism is in this disease will help us to know whether or not for any specific patient those heart muscle cells are actually normal or intrinsically defective. And that's a very important point because in the future there may be some strategies where we actually try to rescue or salvage that left ventricle. To maybe trick it back into growing normally. But if it turns out in a patient those heart muscle cells cannot work properly, then that strategy won't work. So this is just to summarize for you how I started with a human patient, how there were already nice studies in this C-squirt that I showed you in the beginning, and the fruit fly, and that I, along with my collaborators, are continuing to work on fruit fly, another organism that I didn't talk about today called the zebrafish, frogs, these are tadpoles, baby frogs, and mice. And that by combining these different systems, we can learn a lot more than any one system individually. So I want to just spend the last couple minutes talking about some things in the future that I think could be very exciting. As I mentioned before, we know that ETS1 is actually not expressed in cardiac myocytes, right? Which means just by themselves, at least initially, those cardiac myocytes in Jacobsen syndrome may be normal. And as I just mentioned, that suggests that in some cases, there may be a chance to salvage or save that left ventricle. So what I want to show you is some work from a company based in Hong Kong that may allow us to begin to come up with new treatments for hypoplastic left heart syndrome. What you see here is actually a heart chamber that they have made, what they call a heart in a jar. And if you look carefully, can you see how that's contracting? So what this company has done is created a heart in a jar, which I personally never could have imagined this possible. And with this, they can actually do similar studies like what your children have had to look at heart structure and function. And here's where this can be really powerful for our studies. The way they do this is they start with a patient or a normal, and they take a skin biopsy, not a heart biopsy, a skin biopsy. And through some magic genetic tricks, they can reprogram those skin cells to become heart cells. And then they can use those heart cells to build this heart in a jar. So this is obviously from a normal person, skin cells. But what we hope to do in the next, hopefully, year or so is to do that from a couple of my patients with Jacobsen syndrome and hypoplastic left heart syndrome. And in that case, we might predict that those cardiac myocytes by themselves should be normal. Whereas there are certainly other cases of hypoplastic left heart syndrome where we know that those heart cells are not normal. So we will study those patients as well to hopefully show a proof of concept that we can use this system to study further hypoplastic left heart syndrome. Because one of the problems that we see in our patients is some patients develop the right heart failure at a very young age. Whereas other patients, like one I showed you yesterday, is 25 years old and so far is doing very well. But we don't know why, right? So we might be able to apply this system to identify very early on which patients might need more medical therapies or which patients might need to be actually listed for transplant sooner rather than waiting for when it's too late. We can also use this system potentially to discover new drugs that might be able to be used to treat right heart failure. And right heart failure is what usually kills people with hypoplastic left heart syndrome. But it's also a big problem in other people with other diseases like lung disease. And while there have been some advances in treating people with heart failure affecting the left ventricle, there are no drugs that are good for treating the right ventricle when it fails. All right, so that brings me to the end. I want to point out some people from my lab now and before that have been incredibly talented to do this work. So this is Lizu Lin, who unfortunately had to leave my lab recently because she is so good that I can't afford to pay her anymore. But she did all those beautiful studies I showed you with the neural crest. This is Annalyn Schroeder, who actually works in a lab just down the street from me on the fruit fly heart. And she has done those experiments I showed you studying ETS1 in the fruit fly heart. And this is Lu Wang, who is a PhD, what we call postdoctoral fellow in my lab, who is doing all those other experiments that I showed you in the mouse on the endocardium. So this is a list of past and present members of my lab during this time period. Mao Ching Yi is actually back in China, but he was really the critical person to help us discover the ETS1 gene as the heart gene. And then I have a whole number of other great collaborators around the world. Some in San Diego and others in Europe and elsewhere, including, as I mentioned before, where is her name? It better be on here. Shui Ni, who did that incredible experiment in the frog. And as I mentioned yesterday, it's always a challenge, and I've been very fortunate so far to, over the years, have many different sources of funding. Including some of you in this room. And I hope you understand that without that kind of support, there would have been no talk today. And that's it. Thank you. Does anybody want to make a guess where this picture was taken? I'll give you a hint. It's not the United States. Did I hear France? Yeah. More than just France? What part of France? Did I hear Loire? You got it. One of the castle villages in Loire. All right, any questions? Or if I put everybody to sleep?
At the very beginning, you talked about the stages of development of the heart during fetal life. I would like to know what you call early versus late development.
So the question is, what do I mean by early versus late stages of heart development? So I'll explain to you in humans. It's amazing that literally 21 days after conception in the human embryo, the heart starts to beat. 21 days. That's only 3 weeks post-gestation. By about 8 weeks, the basic structure and form of the heart is already done. Meaning the chambers, the 4 chambers, the valves, and the great arteries. So basically, early heart development, in my mind, is all first trimester by up to about 8 weeks. Late heart development, basically after 8 weeks, it's mostly just everything growing, getting bigger along with the rest. And that's, of course, going to be a very big challenge because we don't even diagnose hypoplastic left heart syndrome until later than that. So just as an aside, I'm actually going to have a visiting professor with me in San Diego who is going to be working to try to do earlier fetus imaging of the heart. Because this sounds great, but if we can't even diagnose until way later, then there's nothing we can really do about it if we want to do something like that frog. Does that answer it? Yeah. All right.
I have another one.
Yeah.
Just out of curiosity, I would like to know where is located the Eve gene.
Which chromosome? I should know the answer to that, but I don't off the top of my head. It's not chromosome 11, I can tell you that.
Okay, somewhere else.
Somewhere else. That I can tell you. No more questions. I guess I've answered everything. Any questions from, are there any chat room questions at all? Or? There is. Okay.
Ich habe eine Frage, und zwar, kann sich ein Herzfehler auch erst im Laufe des Lebens entwickeln, wenn ETS1 fehlt?
Is it also possible that a heart problem develops during the course of life when ETS1 is missing?
Did everybody get that okay? Yeah. So, for example, some people are born with just what we call a bicuspid aortic valve.
Manche Leute werden geboren mit einem Bikuspid-Aortenklappe.
And I showed you early on where instead of the normal 3 leaflets, you just have 2 leaflets. Did everybody remember that?
2 und nicht 3 von diesen Blättchen, wisst ihr das noch?
It turns out actually about 1 to 2% of the general population is born with a bicuspid aortic valve.
1 bis 2% der ganzen Bevölkerung ist geboren mit einem Bikuspid.
And that over time, maybe by adult age, that valve develops problems.
Die Klappe kriegt dann Probleme im Laufe des Lebens, wenn man erwachsen ist vielleicht.
And actually, aortic valve surgery is the second most common heart surgery in adults, only behind coronary artery surgery.
Aortenklappe ist die zweithäufigste Herzoperation bei Erwachsenen.
So, for example, I have one young lady in San Diego with Jacobsen syndrome who has a bicuspid aortic valve.
Ich habe eine junge Dame in San Diego mit einer bikuspide Aortenklappe.
That I'm following to make sure that that doesn't become a problem over time.
Und ich folge ihr, sicher zu sein, dass das nicht so ein Problem wird.
So the short answer is yes, problems can happen after birth as well.
Die kurze Antwort ist ja, es kann auch sein, dass Probleme erst nach der Geburt auftreten.
Does that answer the question?
War das eine Antwort zu deiner
Good.
Frage? Gut.
Wie oft sollte man das untersuchen lassen?
How often should one have this checked?
Yeah, I'd say if everything in the beginning is completely normal, probably every 2 or 3 years
Wenn
is
am
reasonable.
Anfang alles völlig normal ist, dann alle 2, 3 Jahre.
Or if the pediatrician, let's say, here's a new heart murmur, that should be looked at right away.
Oder wenn der Kinderarzt ein neues Herzräuschen hört, dann muss man sofort das untersuchen.
Okay?
Thank you.
Any other questions from the chat?
Gibt es noch Fragen aus dem Chat?
All right. Any other questions in the room? All right.
Gibt es noch andere
Break
Fragen
time?
aus dem Chat?
Yeah.
Okay, Zeit
Thank
für
you,
Pause.
guys.
Gut, danke.